For this videos, I will talk about the basic or the important concept that you need to know and also check the workbook with you. So if you haven't done the workbook 4.1 to 4.4, so please, complete it now and post the videos. And if you have already complete, then in this case, you can continue to watch the video and let's go. Okay. So now for here, we can see there is a cell structure. We call it the cell wall. So the basic function for the cell wall, it is help us to protect, support, and give a regular shape towards to the cell. So we can only see the cell wall in the plant cells, but not in the animal cells. And on the other hand, there is another structure that very close to us to the cell wall, and it would be the cell membrane. So cell membrane, it is a structure where both animal cells and also the plant cells do have. And the basic function and the major functions for the cell membrane is to control the movement of substance into and out of the cell. And next, we have the cytoplasm. For cytoplasm, actually, it is some fluid and many chemical reactions will take place here. Both animal cells and also plant cells will not will contain this structure. It is the fluid. So next, for the nucleus, both animal cells and also plant cells, they do have this structure. And basically inside the nucleus, we can see, or we can it is containing what we call the genetic materials. And there is a special case, which is the mature Rappler cells. For mature Rappler cells, there is an exception. It do not contain any nucleus. Next, for the large vacuoles, so it is a membrane bounded sac, and this membrane bounded sac actually it contain or it store water and also dissolved minerals. So inside both animals and also plant cells, they do contain the vacuoles, but for the animal cells, we do not have any large vacuoles. We only have some small vacuoles. Next, for the plant cells, only the green part of the plant cells, they will contain the chloroplast. And inside the chloroplast, it contains chlorophyll. And the basic function for the chloroplast is to undergo what we call photosynthesis. Next, here it will be some structure of the microscope. So the first part that face to us, to our eyes, it will be what we call the eyepiece. Next, the one that for us to turn or to twist the lens, it will be the objectives. Next, and there will be a place for us to place the slide. It will be the one that we call the stage. Next, which is what we call the diaphragm, is allow us to adjust the amount of light entering towards to the objectives. Next, the one that, below, it would be what we call the light source, And then, we have a tube, that containing, or I mean, connecting with the eyepiece, it would be what we call the body tube. Then, the one that will hold the microscope, we call it the arm. Next, the one that for us to adjust, it will be what we call the coarse adjustment knob. It is for us to have a large adjustment. So once we turn it, the stage, it will be moves upward and downward significantly. Next, we also have what we call the fine adjustment knob. And in this case, you seldom see a significant movement of the stage. It just move a little bit for this fine adjustment knob when we turn the fine adjustment knob. Next, we got the base. It is the base for us to place the microscope. So here it will be some description corresponding to different part of the microscope. So as I already mentioned, their basic functions. So in this case, I don't go through with you. So here it will be the answer. And remember, you are required to write down the letter. Next, so how can we do when we see some word? The first thing we need to do, we need to switch, okay, the paper 180-degree clockwise, okay, then in this case, the word that presents to you or is showed to you, it will be the word that it will be shown to you in the microscope. Because for the microscope, most of the image, they are magnified and inverted, okay? They are magnified and inverted. So in this case, based on here, so the answer here, it will be A, okay? Next, Alan is going to observe a leaf under a light microscope. Which of the following combination of eyepiece and also objectives allows him to see the largest number of cells? So largest number of cells. So if you see largest number, meaning that it will be having the lowest magnification. So we need to find the one that having the lowest magnification. Then in this case, it will be C. Next, okay, which of the following statement about light microscope and electron microscope are correct? Now, electron microscope, it will have a higher magnifying power than the light microscope. That is correct, okay? Next, okay, light microscope produce a color image while the electron microscope produce black and white image. This is also correct. Both light microscope and electron microscope are commonly used in school laboratory. Sorry, it is not correct. In our school, we do not have electron microscope, but we have quite a number of light microscopes. So therefore, the answer here, it will be A. Next, the photos on the right show some onion cells. Which of the following about the structure X it is correct? So now we can see X it is in a thickened line. This thickened line actually it indicates that it would be a cell wall. So if it is a cell wall, then we need to look at the description of the cell wall. So is it rigid? Yes. Is it protect the cell? Yes, it is used to protect the cell. Will it present in the animal cell? Sorry, it will not present in the animal cell. So it only presents in the plant cell. So therefore, the answer it will be B. So now let us quickly have a look on the cells, okay, that's or Mary observed it under the microscope. So we can see that there is a thickened line or a broadened, a more darkened line, okay, which shape the edge of the cells. And that actually not any shading, but that would be a structure. We call it as the cell wall. So this would be what we call the cell wall. As there is cell wall, as there is the presence of cell wall. So in this case, okay, the cells that Mary observed, it would be the plant cell. Okay, plant cells. So give two reasons to support your answer in A. First, okay, we can see that there is presence of cell wall. Apart from that, there is something special. There is some green colored structure. Now, this green colored structure will be, the green color structure will be the chloroplast. So in this case, okay, based on the reasons or the photograph, photograph. So in this case, okay, the why? Okay, we will say, okay, it is a plant cell. This is because the cell, okay, contains cell wall. The cells have the chloroplast, or the cells have a regular shape. So any two of them, it would be fine. So which of the eyepiece and also objectives is Mary using to observe the cell? So in here, we can see that the magnification is 400 times. So in this case, what we need to do is that we need to find out the combination that can give you 400 times. So in this case, it only have 10 times here and 40 times here. So the only options that in order for Mary to get 400 times, the eyepiece must be 10 times, while the objectives, it would be 40 times. So if Mary wants to make the following changes. He want to brighten the field of view. Then in this case, how can she do? So in this case, first step, he need to adjust the diaphragm. Or on the other hand, she need to turn, okay, or with a brighter light source. It is both are correct, okay? Then how can we move the cell X to the center of the field of view? Now this is cell X. So if I would like it to in the center, so it need to move to the right hand side first and then move to the um, downward. So if it requires move to the right hand side and also downward, and we notice that actually the image is inverted. So we need to write it in reverse. First, it need to turn to left. Okay, move the slide to the left. And what? Move the slide towards to up. So why? Because as it is inverted. So what we need to do, we need to turn our paper, okay, 180 degrees Celsius. Then we can see in order for us to make it or turns to the correct direction, we need to turns to the left and turns to upward, move the slide upward. Is it okay for that? Okay, next, okay, so here it will be the drawing of the cells. So how can we draw? Now remember, first things, okay, we need to use the pencil to draw it. So in order for us to use the pencil to draw it, okay, you need to bring the pencil. So then, okay, the second step, we need to sketch, okay, use the pencil to sketch, okay, the structure, okay, of the desired cell that we would like to draw. Next, okay, there is a nucleus, and you can see there are some dots like in between the nucleus and also the cell. So in this case, these are actually some what we call some stain, okay, or maybe some bubbles. So you no need to draw those parts. So in this case, okay, we can see that actually it is a human cheek cell. So therefore, it is an animal cell, so they do not have any cell wall. So it would be the cell membrane. Next, okay, the part that in between, okay, the cell, it would be what we call the cytoplasm. And last one, okay, for us, okay, to see, okay, it is a circle, it would be what we call the nucleus. Then is it finished? No, remember, you need to write down a title. Now, for us to write down a title, so it is 400 times. It is 400 times if, okay, if the magnification is higher than 100 times, then it would be a high powered labeled diagram. Okay, so in this case, okay, we can see that it is 400 times. So in this case, it would be a high power label diagram of a human cheek cell. Sorry, just a second, because not enough space, okay. A high power label diagram of human cheek cell. Then remember, you need to underline, okay? But is it finished? No, there is one more thing. You need to write down the magnification. The magnification in here, it will be 400 times. So if we place the magnification at the back, it is required for us to write down the capital letter, capital letter. So in this case, it would be 400 times. Then, okay, so we move on to the next part. Say one safety precaution that we should be taking when we handle the plant or the animal experiment. We need to wear the disposable gloves, or we need to wear the safety spectacles. Next, okay, why we need to add iodine solution, or why we need to add okay some solution into it. So these solution actually is serve as what we call staining. Okay, stain the cell. Okay, so that we can observe it clearly. Next, okay, so why it is important for us to cover the edge of the cover slip first before, okay, we drop the cover slip. This is because without we cover, or we place one edge of the cover slip first, then there will be a lot of air bubble being trapped inside to the slide. So therefore, to prevent the air bubble from being trapped, it is important for us to place, okay, the slide, okay, something like that, okay. This is the slide, and this is the cover slip. Touch the liquid first, and then slowly attach on it, okay. So this is what we call a method for us to reduce the number of gas bubble or the air bubbles. Next, okay, why we will just use the water rather than iodine solution when we are added towards to the leaf. This is because leaves contain chlorophyll, so we don't need to stain the cell because it already got the color. So now we can see that, okay, if the magnification it is at 20 times, then in this case, which of the following sets of IPs, okay, is appropriate, okay, to be used? So in this case, it will be IPs containing 5 times, and while the objectives, it will be 4 times. So next, okay, we'll move on to the 4.2, okay, from a single cells to a multicellular organisms. So normally we will say that new cells are formed by a process, what we call the cell division. Next, by undergoing cell differentiation, the cells will become specialized for a particular function. Now remember, for cell differentiation, there is no any increased number of cells. Okay, there is no any increased number of cells. But for cell division, okay, there is increased number of cells. So next, okay, DNA determines the trails, okay, what we call the trails. Okay, trails here actually it referring to the characteristic or some appearances, okay, of a living things. So it coiled tightly to form what we call a structure of chromosome, okay, chromosome. So during the cell division, okay, so the chromosome will be, I mean, the DNA will coil it tightly and shorten it, okay, and form what we call the chromosome. So here it will be the order of the cell division. Now for this part, no need to work on it. Okay, in the upcoming examination, it will not be covered. It will not be covered. Okay. So next, in the process of cell division, which of the following does not occur? The genetic materials in the cells make a copy of themselves. So it should be occur, otherwise it will keep having less and less genetic materials once we do the cell division. Next, the cytoplasm will divide into two. Cell membrane separate and cell membrane become thickened. Now remember, cell membrane, it will separate. Cytoplasm also will divide into two, but the cell membrane will not become thickened. So in this case, okay, D, it will be the wrong answer. Okay. So next, which of the following statement about the DNA in human cells are correct? So it is a type of genetic materials. Yes, it is correct. It is present in the cytoplasm, so it should not present in the cytoplasm. It should present in the nucleus. Next, its structure look like a twisted ladder. So in this case, it will be correct because it is double helix. Once it is double helix, it is similar to what we call a twisted ladder. It is a double helix for the DNA. So the DNA, it is double helix. Double helix, okay? So therefore, okay, so the genetic material, it is stored inside what we call the nucleus, okay, the nucleus. And then, okay, the genetic materials are organized as what we call an X shape, and we call this X shape as chromosome. So next, okay, this chromosome actually is made by a long coil of DNA. And for the DNA, the full name here, it will be deoxyribonucleic acid. And there are four kinds of bases, okay? And each segment, we will call it as a gene, okay? So for the B and C part here, actually it is an extension, so I don't go through with you. So next, okay, for the sex cell and also the reproductive system, the human sex cells are called the sperms, okay? So the head contain what we call the nucleus, and the tails, okay, it will beat, okay, to allow swimming. Next, human female sex cells, okay, are called what we call the ova or the ovum. Next, the cell membrane is surrounded by what we call a jelly coat. This jelly coat is not a cell membrane. It is some other things different from cell membrane. You can think it is some uh, extra substance just deposit on the cell membrane. Next, okay, the cytoplasm contain a lot of food substance for early embryo development. Next, both male and female sex cell, it contains 23 chromosomes. Next, okay, the X chromosome cannot be found in human sperms. Sorry, it is false, okay? Both X and Y chromosome can be found in human sperm because sperm actually is coming from male, and male, it has the possibility to give the gamete cells containing X chromosome or Y chromosome. Next, an ovum carry half of the number of this chromosome found in the body cells. Yes, it is correct, true. Because it is a gamete cell, it is a sex cell, so therefore it will only contain half of the number. Next, human zygote, it carry 46 chromosomes. So sorry, it will carry 23 chromosomes. Sorry, it is false. Why? Because it should be contain 46 because it already forms the combination. Because zygote, it is the combination of sperm and ovum cell. Is it all right? Next, okay, here it would be the structure of the reproductive system. So I don't go through with you. And the part that cover, okay, or to hold the testis, we call it the scrotum, okay, scrotum. And this would be the female reproductive system. And I don't go through with you because you can just directly found in the textbook. So next, okay, for the matching, okay, so it would be CADB. Okay, so you can just directly found in the textbook. So I also don't go through with you. Next, okay, so we can see here, okay, the statement about the human ovum are correct. So in this case, okay, there are quite a number of things that we need to know. So now let us quickly have a look. Okay, so for the ovum are correct. So it is much larger than a sperm. Yes, it is correct. Okay. Next, it has 23 pairs of chromosome. It is not correct. Why? Because it is 23 pairs. 23 pairs meaning that there is total of 46 chromosome. As ovum, it is a sex cell. Okay, it is a sex cell. So in this case, okay, it should be only containing 23 chromosome. So in this case, there should not be 23 pairs. It's only 23 chromosome. Okay. If it is 23 pairs, then meaning that it would be equals to 46. So it cannot move by itself. Yes, it is also correct. So therefore, the answer here, it would be 1 and 3. Next, okay, if you look at here, we got how many chromosomes? We got 46 chromosomes in here. So meaning that it is a body cell. It is a body cell. So in here, it would be, must be correct. So the cells, it belongs to female. So it is, of course, it is correct because X and X. Next, it is belongs to an adult. Can we see whether it is belongs to an adult or it is from a children? No, we can't because the chromosome, it cannot tell us which stage it is. Okay. It just tells us whether we are a human or we are not a human. We are male or we are female. Okay, so in this case, it would be not correct. Okay, next, is it the cells it is obtained from the liver? We don't know. Okay, it can be from other organ. Okay, but it is definitely it is from a body cell, some cells of the organ. So therefore, here it would be A1. Okay. So next, okay, human sperm and human ovum, they both have the same amount of cytoplasm. Of course, it is not correct because the ovum is a lot larger. Next, they have same amount of food substance. No, of course not correct because the ovum, it has more amounts of food substance. So they have same number of chromosomes. So do they have same number of chromosomes? Yes, they do have the same number of chromosomes. But do they have same genetic materials? No. Why they do not have same genetic materials? Because, okay, some of the ovum, they may carry, okay, X chromosome. But some of the sperm, actually, they may carry Y chromosome. So in this case, okay, the genetic materials is different. So therefore, the answer here, it would be C. Next, which of the following structures in males, okay, is or are involved in the production of semen? So testes, yes. It is sex glands, yes. And how about the penis? No. Penis, it will not produce any fluid for the semen. So it would be only 1 and 2, which is B. Next, okay, we move on to 4.4. So the signs of sexual maturity, boys may experience wet dreams, and while the girls will start to have what we call menstrual cycle. Now, after the sexual intercourse, okay, the sperms in the vagina will swim upward and through the uterus towards to what we call the oviducts, and then the sperm may fuse with an ovum to form a zygote, and this process we call it fertilization. So when the embryos reach the uterus, it attaches towards to the second uterine lining, and this process we call it implantation. In the uterus, the embryo grows inside a bag, we call it the amnion, and about 8 weeks after the fertilization, the embryos will develop into a what we call the fetus, okay, with all major organs are formed. Next, okay, the growth of the hair in the pubic area and also the armpits is a secondary sexual characteristic in both boys and girls. Yes, it is true. Okay. Next, not every boys have wet dreams. Yes, this is also true. The length of menstrual cycle is always 28 days. So sorry, no, some may be longer, some may be short term. For fertilization, it occurs in oviducts. Okay, so sorry, it is sorry, the fertilization occur in vagina. Sorry, it is not occur in vagina. So it should be occur in oviduct. Okay. Next, okay, in females, in human females, embryos normally develop in the vagina. Sorry, it is also not correct. Okay, the fetus, it should be developed in the uterus. Uterus. Okay. It should be uterus. Okay, next. Then, okay, so for the multiple choice, okay, so which of the following are the secondary characteristic? Okay, production of sperm is not correct. Shoulder, okay, it is a what we call a sign of maturity. The sign of maturity is different from the secondary sexual characteristic. Secondary sexual characteristic is some other factors for us to define you are which gender. So in this case, shoulder become wider, beard grows on the face. So it would be the example for us to have, okay, as a example of the secondary sexual characteristic. Which of the following statement about menstrual cycle is correct? Menstrual cycle usually lasts about 28 days, not always, okay. Next, okay, ovulation occurs during the menstrual cycle. Yes, okay, of ovulation, it should not be occurs during the menstrual cycle. It should be occur after the menstrual cycle. Menstruation. Now, for menstruation, okay, it is talking about day 1 to day 5. And ovulation, we are talking about day 14. So in this case, this is not correct, okay? After menstruation, uterine lining will thicken again. So this is correct. So therefore, the answer it will be A. Next, during sexual intercourse, the semen discharge into the vagina. Remember, okay, for the boys, okay, we would not discharge any semen directly towards to the uterus, okay. So next, okay, when girls enter the puberty, okay, which structure will start to have the periodic changes, okay. So definitely it will be 2 and 3. So why it would be 2 and 3? 4, okay, 2, it will be occurring what we call the ovulation, in which, okay, there will be a release of ovum. And for 3, okay, there will be entering what we call the menstrual cycle. And for the menstrual cycle, there will be what we call the thickening of uterine lining. So in this case, this will change a little bit in the thickness, okay? And this is why there will be a periodic changes. Okay, next, which of the following statement about placenta in human is incorrect? Okay, so it should be D. Okay, it will not contract strongly during the childbirth, in which we have go through during the lesson. Which of the following may happen during the birth process? Okay, amnion will break, the neck of uterus will become wider, and the muscle of the uterus will try to do the contract, and the contraction is very great and vigorous, and in this case, this will push the fetus or push the baby out of the body. Okay, so therefore it would be D. So in this case, okay, which of the following periods it is menstruation most likely to occur? Menstruations to occur, then in this case, there will be a drop in uterine, the thickness of the uterine lining. Okay, the thickness of uterine lining. So when it will having the drop of uterine lining, so in this case, it will be starting from 1st of May towards to 5th of May. So in this case, this would be the answer. Next, complete the paragraph below. When the ovum release is not fertilized, then in this case, the second uterine lining will be continue to grow. That means it will be thickened again. It will be thickened again. Then, okay, once, okay, it reached day 28, so it will enter a new menstrual cycle. Then in this case, the thickened uterine lining, it will be broke down, okay? And in this case, menstruation will occur. So the tissue of the thickened uterine lining and unfertilized ovum and some blood, okay, would then pass out of the body through what we call the vagina, and this is what we call the menstruation. So suggest the days when the ovulation most likely to occur. So if 1st of May, it is the first day, then we count for 14 days. So in this case, it will be 14th of May, it will be the first day for the ovulation, okay? Next, okay, it's not the first day, I mean the date of ovulation. Next, okay, for the structure of A to D, okay, A, actually, it will be what we call the placenta, okay? And then, okay, B, it will be the umbilical cord. Next, C, it will be the outer part. Then that would be the uterus, the muscle, the uterine muscle. And D, it will be what we call the amniotic fluid that enclosed by what we call the amnion. Okay, so which structure it will protect the fetus against shock? Remember, the amnion, it will not prevent or it will not protect the fetus against shock. Okay, the amnion, it will not protect the fetus against shock. Okay. Only the amniotic fluid will prevent or protect the fetus against shock. So in this case, what is the major functions of structure A? So for structure A, placenta, the major function is through structure A, the fetus can obtain nutrient and also oxygen from the mother's blood. And at the same time, waste was passed from the fetus towards to the mother's blood for removal. So it is an exchange of blood. And for the fetus, they will obtain the nutrient and also oxygen from the mother's blood. But the waste, from the fetus, for example, the carbon dioxide and also some waste, through the metabolic reaction will be passed towards to the mother's blood for removal. So what happens to structure A after the fetus it will born? It will detach from the uterus, so it will be pushed out of the mother's blood, mother's body. So that would be the whole debriefing on workbook 4.1 to 4.4. So again, if you have any other questions, you can just ask me via the Google Classroom.